the way we're going to approach this is we're going to go back to the source. All right? We're going to go back to the source of the basic laws that govern inheritance. And we're going to talk about Gregor Mendel, who was a monk in the 1800s, who discovered, essentially on his own, the laws that govern inheritance, though they weren't rediscovered and actually became part of science for another 50 years or so, 40 years, 50 years. But we're going to think about his experiments, the context that they occurred in, the results he got, and the interpretation that he gave for the results. And once we've done that, we're going to then take that and try to apply modern genetic language to it. And so what I've given you guys on this worksheet, wherever it is a list of terms to define. And so we're going to go through these and define them as we put Mendel's discoveries into modern genetics language. But I would say it will help you a lot to understand the material on Monday if you go through and try to define these terms and understand them yourself before we discuss it on Monday. Okay? So to sort of the setup for how we're going to approach this make sense, we'll talk about it in the historical context, then we'll apply modern genetics language to it, and then we're going to look at meiosis and see how meiosis actually causes these patterns of inheritance that Mendel discovered. So we're going to kind of tie it all together. So Gregor Mendel, here he is. Normally, I have this little bust of him that I can like take and show you guys, but we don't have it in this room, so I can't show that to you. It's kind of interesting to think about Mendel and Mendel's work because Mendel worked in an abbey, so he was a monk, and he was also somebody who pursued science. And I think it's interesting to kind of reflect on what now there's a huge kind of divide in a lot of people's mind between religion and science, and it's like they're incompatible in some way in some people's mind. And I think it's kind of cool to look back at people who were really leading major discoveries in science and seeing that they actually were also leading members of the religious community at the same time. And obviously, you know, there's a whole bunch of stuff going on socially, culturally, religiously, in terms of science as well. But it's just kind of interesting to reflect on how different it can be in different times. So Mendel worked on the pea plant. And this is the mighty pea plant. It might seem like an unlikely candidate to discover all of the rules that govern inheritance. But it turns out it's actually a really good model organism for doing breeding experiments. And the reason it's a good model organism for doing breeding experiments there's several reasons. One is that pea plants already, at the time of Mendel's work, had been bred to have many, many different traits in them. So people were already really interested in peas, and they bred peas because people love diversity. And so we love having variety in our food, variety in our flowers, variety in everything. And so people had already bred into existence all of these different varieties of peas. And so people had, at the time of Mendel's work, peas that were purple flowers and white flowers, peas that had yellow seeds and green seeds, peas that were round or wrinkled, inflated pods or constricted around the seeds, green pods or yellow pods, short stems, tall stems, ones with the flowers at the tips or ones that have flowers coming at the axis where a leaf joins the stem. So these were all traits that people had bred into existence. The other cool thing about pea plants is that they have a pretty fast generation time, which means that you can, within a single growing season, do multiple crosses. So you can cross, collect the seeds, plant, and do crosses, more than one cross. And I don't know if anybody grows sweet peas. Anybody? They're really fast. Like you plant them and like 3 weeks later, they're setting seed. So it's potential to do multiple crosses in a single generation, as opposed to some organisms where it's one per year or one per 5 years, right? Or one per 20 years. So it's a pretty good organism for that. The other thing that's nice about it is that pea plants are both male and female. So you have the ability to sort of cross between individuals in a very, very easy way. So you can take pollen from one plant, put it onto the female part of the other plant and pollinate it, and do the reciprocal crosses very readily. So that's a cool thing about pea plants, and one of the reasons that they're a really useful organism in this type of study. So what was known at the time of Mendel's work was when you did certain crosses between peas that had certain traits, there were patterns that came out of it. And one of the patterns that was known, in my understanding at least, before Mendel did any of his work, was that if you take purple, for instance, flowered pea plants and you cross them to white flowered pea plants, all that you got was purple flowers in the offspring. And it was even known that if you took those purple flowers of that second generation and you crossed them together, what would pop up in the next generation would be a mix of purple and white. So this was something that it wasn't a discovery of Mendel's necessarily. It was something that was already known because, well, people have been breeding these things for quite a bit of time. It was even known, at least my understanding, was that when you did these crosses, what came out in this next generation was a preponderance of purple, meaning more purple than white. So purple crossed to white gives purple. These purples crossed together give you a mix of purple and white, and more purple than white. But what Mendel did that was different than a lot of people is that he was a very, very rigorous and careful scientist. And when he did these crosses, he counted very, very carefully the number and type of every individual that emerged from the crosses. And so I'm going to give you some of his data, and I want you guys to look and see if you can see any patterns to the data.